Glycolysis is the first step in the breakdown of glucose to extract energy for cellular metabolism. This takes place in the cytoplasm of a cell. Glycolysis converts one 6-carbon molecule of glucose into two 3-carbon molecules of pyruvate. This makes 4 ATP but uses 2, leaving the net product of 2 ATP and 2 molecules of NADH.
Pyruvate oxidation links glycolysis to the rest of cellular respiration. It's an aerobic process because it needs oxygen. Pyruvate oxidation is how pyruvate from glycolysis is made into acetylcholine, so it can enter the Krebs cycle. The pyruvate enters the mitochondrion to get to the mitochondrial matrix. The first step is our carboxyl group is snipped off the pyruvate and is released as CO2. This 2-carbon molecule is oxidized and/or reduced. The electrons lost are picked up by NAD+ to form NADH. The oxidized 2-carbon molecule is attached to the coenzyme A to form acetylcholine. This carrier molecule carries the acetyl group to the Krebs cycle.
The Krebs cycle, aka the citric acid cycle, is a central driver of cellular respiration. It takes acetylcholine, which was produced by the oxidation of pyruvate, and, in a series of redox reactions, harvests much of its bond energy in the form of NADH, FADH2, and ATP molecules. In step 1 of the Krebs cycle, acetylcholine combines with oxaloacetate to form citrate. CO2 is released in similar reactions that make NADH each time. In step 2 of the Krebs cycle, the remaining 4 carbon molecules undergo a series of reactions that make an ATP, then reduce FAD to FADH2, and makes another NADH.
Oxidative phosphorylation is the metabolic pathway in which cells use enzymes to oxidize nutrients. The electron transport chain forms a proton gradient across the inner mitochondrial membrane. This drives the synthesis of ATP via chemiosmosis, which is the movement of ions across the membrane. Step 1. NADH and FADH2 transfer their electrons to molecules near the beginning of the ETC. Step 2. Electrons are passed down the chain from highest to lowest energy. Step 3. At the end of ETC, electrons are transferred to oxygen and pick up a hydrogen ion, which makes water. Step 4. As hydrogen ions flow down the gradient, ATP synthase, which harnesses the flow of protons to synthesize ATP.
Fermentation and anaerobic respiration are how cells extract energy from glucose without oxygen. Anaerobic reactions in muscles make lactic acid. Fermentation starts with glycolysis, but extra reactions have to regenerate NAD+ from NADH.
Alcohol fermentation is when NADH donates its electrons to a derivative of pyruvate. This produces ethanol. In this process, a carboxyl group is removed from pyruvate and releases carbon dioxide, which makes acetaldehyde. NADH then passes its electrons to acetaldehyde. This regenerates NAD+ and forms ethanol.